Actinomycin D. To begin this informational video, we will briefly discuss the fundamentals of DNA and its function. Then, we will explore the background of actinomycin D and the mechanisms through which it works to inhibit cell growth within a tumor. DNA, in its natural form, exists as a two-stranded structure known as a double helix. It is composed of 4 nucleic bases: guanine, cytosine, adenine, and thymine. Illustrated in the diagram to the left, guanine is bound to cytosine and adenine is bound to thymine within the double helix. Here we have the process of how DNA is used to create proteins within the cells. The double-stranded DNA is used as a template to form a single-stranded molecule known as RNA. From here, proteins already present within the cell act on the single-stranded RNA to manufacture proteins that are needed to keep the cell alive. This diagram represents the cell cycle of a normal cell within your body. The blue arrows represent times at which a cell is using DNA to make RNA so that it can synthesize necessary proteins. The red arrow indicates a period of time in the cell cycle where one cell is dividing to 2 cells through a process known as mitosis. In normal cells, this is heavily regulated. In tumor cells, this process is left unchecked. What makes a tumor grow? In normal tissue of the body, a cell receives signals from surrounding cells indicating that it does not need to divide. This is known as contact inhibition. For cells that do not receive this signal, they must pass a series of checkpoints before they can divide. In the cancer cells, there is no contact inhibition. Cells are not signaled that they are not to grow or divide, which results in the formation of a tumor. Additionally, the abnormalities within the cells allow for unregulated cell divisions. The illustration depicts the difference in growth seen in regular cells compared to those of cancer cells. Normal cells maintain a sense of regularity, while the cells of the tumor are unordered. Actinomycin D belongs to categories of drugs known as anti-tumor antibiotics. Anti-tumor antibiotics are produced by Streptomyces, a type of soil fungus. Actinomycin D belongs to the subcategory called chromomycin. It is used to treat tumors due to the way in which it interacts with the DNA of dividing cells. The main mechanism of actinomycin D is to insert itself into the double-stranded DNA molecule in specific regions. To the right of the screen, the drug molecule, which is shown in blue and green, can be seen entering the double-stranded DNA. The specific regions that are targeted by the drug are those that are rich in guanine and cysteine bases, identified by the boxes in the diagram to the right. In addition to inserting itself within double-stranded DNA, actinomycin D targets single-stranded DNA rich in guanine and cytosine. In this case, the binding of the drug to the structure to the right, which is known as a hairpin turn, results in the single strand breaking and not being able to function properly. After the drug has inserted itself within the DNA, it interrupts the function of RNA polymerase. This is the enzyme that is responsible for transcribing DNA to RNA within the cell. Without being able to transcribe DNA, the cell is not able to produce the necessary proteins to both survive and divide. Now I'm sure you're wondering why these specific regions of DNA are more important than others in terms of actinomycin D interactions. The main reason is the presence of guanine, cytosine-rich regions being present within the promoting regions of certain genes. The promoting region is important to signal where the DNA needs to be copied for a particular gene. These specific genes allow for cancer cells to increase in numbers at a much quicker rate than normal. Normally, the enzyme needed to make a single-stranded RNA is signaled by the promoter region to bind to the DNA as shown on the left. With actinomycin D bound to the promoter region of the DNA as shown to the right, the enzyme needed to make single-stranded RNA is not signaled to bind to the DNA, preventing the gene from being normally expressed. Due to its effects on DNA and the inhibition of certain enzymes, actinomycin D causes cells to undergo programmed cell death, known as apoptosis. This is a stepwise process in which the cell shrinks in volume, the membrane becomes distorted, and then breaks the cell into smaller units. These smaller units are then broken down by the white blood cells within your body.